Next one is the multiple sequence alignment. So, we discussed three different methods. One is a statistical method, Chauffasman, and we discussed about information theory, GAR method, and the heterogeneity profiles, right, based on the patterns in heterogeneity. These are based on single sequence. If we have one sequence, we do not need anything else, just see the values, the preference of residues or the profiles we can predict. Later on, we realized that instead of using a single sequence, if you obtain the information similar to that particular sequence, that may increase the prediction performance. So, they started to use the multiple sequence alignment, right. How to get the multiple sequence alignment? Take your sequence, get the homologous sequences and you can align, right. What program we use to get the multiple sequence alignment? Cluster W, right. Cluster W, cluster omega we can use for the multiple sequence alignment. So, for example, this is your sequence. You obtain the homologous sequences and then you can get this multiple sequence alignment, right. Now, you have several sequences and finally, you can see multiple sequence alignment. And if you look into the sequence, some of them are conserved. What is the conservation? Occurrence of same residue at same position, right. For example, take the first residue, it is highly conserved, and some cases it is variable. This is P is conserved here and Y is conserved here, and some positions you can see the variability, right, changes with several residues. For example, you take here I, K, V, right, and yes, there is very many residues, right, they change the variability. So, if it is highly conserved, then the possibility of the prediction performance is high, right, because we take the same from different sequences. So, what to do when you make this sequence alignment, right, we have the multiple sequence alignment, there are different ways to do. First case is we can get the GAR values, right, that means we have the frequency of occurrence matrices. So, get the frequency of occurrence, right, for the helix, strand and coil for the different residues in the sequence alignment, and then you can see which one is the highest one, we can predict. Here the confidence increases if it is highly conserved. Why? Because we get the same information for the same sequence, right. In that case, you can, if you add up all these things, you will get a high value. So, if it is highly conserved, you will get the high confident values. So, if it is insertion deletion, where there is a gap, then we use a value 0, and others they get the table for the helix, strand, coil and turn and get the values, and finally look at for the 4 different states, then you can predict. If the confidence is very high, based on the conservation score, right, between 0 and 1, if it is 1, then you can get the high confidence than the value of 0. So, the first predict is Z-pred. This is the first secondary predictor using multiple sequence alignment, using this one. Then there is another way to use a multiple sequence alignment for predicting the secondary structures, right. For example, right, if we have this multiple sequence alignment, this is unknown, and for example, if you get these numbers from known, that means you search your own sequence with BLAST with the data set of PDB sequences, right, with the known structure sequences, and if you could do this alignment, the first one is unknown and all other sequences we know the secondary structures. For example, we take a second sequence, we change this sequence in a secondary structure, right, that means the sequence will be, right, for example, if you have the sequence, this is your unknown sequence. So, you align with the known values, known structures. Then if you have the different alignment, aligned sequences, these are the aligned sequences. And for each aligned sequence, you have a secondary structure assignment. For example, this is helix, so for each cases you have the secondary structure assignment, and see the values here. If it is helix, 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 then you can assign this value for this. So, for each positions, if you know have the known secondary structures, you can assign the values and then you can get the secondary structure for your query sequence. If the accuracy is high, you can see the conservation is very high. Other way around, if the conservation is high, for example, if these highly same residues, and this case you can see the prediction values with the high accuracy. So, the alignment score increases or the multiple sequence alignment is highly confident and then the accuracy increases. So, they did for several proteins with the homologous sequences and try to evaluate the performance and see the performance, which are the sequences which are highly aligned, they have higher performance than the residues which are in the lower performance, right. This is currently in the research problem, we have the issues the disorder region, because it is very difficult to get the proper alignment. So, it is very difficult to predict these secondary structures when they form with any of the complexes. So, now there is the method called JPRED. This is the one of the earliest methods. They used the multiple sequence alignment. It takes the any of these amino acid sequence, the sequence, and if you make prediction, right, this will give you the sequence alignment for any given sequence, right, as well as secondary structure, right. For example, if you run your own sequence, so this is your multiple sequence alignment and here you can see the predicted method. Okay, here this stands for helix, this arrow stands for strand. So, you can see the multiple sequence alignment, several residues are highly conserved, and if the conservation score is high, then you can see the prediction accuracy is also very high, they can get this information. So, we discussed about the propensity and the information theory, hydrophobicity profiles and the multiple sequence alignment, right. If you see the performance, each method you can see improvement of performance, right. First we started with 60%, 65%, 68% and the multiple sequence alignment, if you get good alignment you can get up to 65 to 70%. So, now they try to put all the information in machine learning, right, because here we try to understand, right, which feature is important, how far each feature performs in the second second prediction. So, you add all the information and put in machine learning techniques, right. Machine learning techniques is just the mapping of the input features with the output. For example, if you have 100 proteins, all the 100 proteins we know the assignment, segment 5 to 10 is helix, 8 to 12 is strand and so on, and here we have the information, central residue we know, neighboring residues we know, patterns we know, preferred residues we know, we give all the information. Then the machine learning is a kind of black box, it maps this input sequence with the output and assign weights to maximize the performance, right. In the later classes we will discuss more details. So, they give the information regarding the sequence alignment, right, for the evaluation information and they use any of the machine learning techniques. There are several techniques available, neural network, support vector machines, right. So, many methods available. So, for example, if you take the neural networks, it learned the known information, that is what we give as input, right. For example, amino acid pattern or any neighboring residue information, and they try to learn this information. They will also use the different windows, they can use 3, 5, 7, 9 and then optimize the windows and optimize the parameters or any properties to predict the secondary structures. How it does? So, take the input sequence, this is your protein sequence, for example, if we have this K, T, L, V, M, T, S, A, V, then we get the profile matrix, right. How to get the profile matrix? PSSM, right, you get the multiple sequence alignment, from the multiple sequence alignment you have the preferred residues at any particular position, so we get the values. Then they take the window length, right, they can use different window lengths, right. So, for example, if there is 13, there is minus 6 to plus 6, right, all the information they take and put everything in the machine learning, for example, neural networks, it will give you the probability of each residue to be in helix or in strand or in coil. So, different, for example, if you take 3 states, right, it will give you the probability, if this is the case, okay, central residue 3 1 in, okay, these are the neighboring residues and these are the preferred residues and so on. It can map, so that this probability of helix is 0.5, strand is 0.2 and coil is 0.1, right. Then we can see from the voting you can see the best one, this could be in the helix or strand or coil and so on. So, here this is an example. So, here you have the input sequence. So, this is central residue, this is Y, right, the input layer they have the 21 values, 20 values for the 20 different amino acids, right, one for the padding sequence, let them align the next one, or they can also use it for insertion deletions. So, Y is here, right, so they put 1 here, right, and all others that is 0. Likewise, then they have some hidden layers, here is a place they train your data. So, they assign different weights, right. So, put either here or here, right, different weights they do between this I and j, right, and based on these weights they give the values for the output. What is the preference or the preferred probability of that particular residue y in helix or in the strand or in coil. For example, if you give this 1, this is a probability 0, this is probability 0, then we can say this residue L is in helix. So, likewise they do it for each, not residue L, this residue is, for example, if it is y, then this is, you can see this as y, right. For each residue we can get the probability, from this probability you can see whether this can be helix or strand or coil. So, this is the server, right. So, it takes the amino acid sequence, right, as an input, right, and it takes the information that you obtain from the sequence, then they give the desired output, right. This is your sequence, okay, and here you can see this is your secondary structure. So, the PSD is one of the highest performing methods, right. So, it could achieve an accuracy of about 70% at that time in 1990s, right, 1993, right. They use the neural network to map the information, right, and to predict the secondary structures at highest accuracy. So, now if you see the growth from the statistical methods to the machine learning, right, you can see the growth in the accuracy, and finally they could be able to combine different information in the neural network to get the highest accuracy. Now, the next one is, okay, so they have different methods, and whatever the methods you take, right, the performance is around 60 to 70%. So, now they try to use the consensus. For example, if you have 10-15 prediction methods, take any amino acid sequence, try to use different methods and predict the secondary structures, and see are there any tendency of different methods to predict the same residue in same secondary structure. In this case, they take a particular sequence, right. For example, if we have one sequence here, use a Javan-Fastman and you get the values, get the secondary structures. For example, this is E, 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 H, 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 C, 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 H, 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 H. Then we use GAR and we get the secondary structures, right. For example, it will give you like this. Then we use a profile and multiple sequence alignment, right, and we use the neural network, we get the values. Then compare these values, for example, this region, if all methods could predict helix or majority of voting, if for example, if you have 11 methods and if 8 methods predicts a particular structure like helix or strand, then we can assign that residue prefers to be in helix or strand, right. So, now we can compare the experimental data with the known database, right, and see how far we can improve. So, if we do this consensus method or the joint prediction methods, they found that this method could improve the performance compared with any individual methods, right. This is one. And the second aspect, they can also do the output of each sequence, right, we will get the secondary structures, right, and this can be the input of any machine learning. For example, if you take this can be the input of information, again they put this also input information. Instead of giving the sequence, they give the output obtained from the sequence and they train and see what is the possibility of having helix or strand or coil. Then the method will train and see the first residue could have the probability of helix or strand or coil, right. This ways we can do. So, how to do this? First you can see any sequence, okay, this is your input sequence, right, and they use different methods. For example, JPRED is based on what? Multiple sequence alignment, right. They also later on use the other information, right, for example, the machine learning and all, right. So, mainly is developed initially based on multiple sequence alignment. So, JPROD predicts this is a helix, the helical regions. Then take the GAR. What is GAR? Information theory, right? Based on information theory. So, they predicted this as beta strand, and here this is the helix, right? Some coil, right? And the whole segment they predicted as helix or strand or coil. And this another method PhD, right? This is based on neural networks. So, this is the values. Here for example, I show only 3 methods. Currently several methods are available, and you can use as many methods as possible depending upon the performance, right? And then see, are there any consensus? For example, this region, right? For this region, if you see, all the methods predict as helix. So, there is no conflict. So, you can use this region as helix. And this region, if you see this region, so here this predicts as helix, right? This predicts as helix, but this predicts as coil. But if you take the majority of voting, out of 3, 2 predicted as helix, so we can take this as helix. But we see the confidence, okay, this may be predicted the higher confidence than this region, because here the probability is less compared with this region, right, this helical region. Likewise, if we see here, you can see this predicted as E, so this also assigned as E. Some cases is totally different, this as helix and this predicted as coil, and this predicts extended state beta strand. In this case, we do not know which one is this one is confusing. Here we use only 3 cases, this is why it is confusing. If you use 10 or 11 methods, then you will have some sort of numbers, and based on the numbers we can assign the secondary structures and see which segment can be correctly predicted, right, compared to the experimental data. So, this is a complete sequence, and we will get the values, and this method is called the consensus, or you can see this is a joint prediction, right, or the based on this voting, the ensemble based method. Second one, we can use the meta prediction. There are several methods available. So, what they do, they first take a sequence. For example, if we take the sequence and predict the secondary structures, they use this information, right, as the input and train this data using any machine learning, and you get the output. In this case, it will consider this type of situations, and they try to maximize the results, because we know the experimental data, right, to see how to assign the values. These meta predictions usually works better than other methods, any other individual methods which are reported in the literature. So, there are several methods which can individually predict. The main advantage of individual prediction method is we can explain why the method predicts well, why the method fails, because if the propensity is high and the region is beta strand, right, we know that this is high values, because of that high value it is wrongly predicted. Even if it is high values and if it is helix, then we can say here high propensity, this is the reason it could correctly identify the helix. Likewise, the information theory or the profiles we can explain. When you go with the machine learning and go with these meta predictions, we get better results, but the problem is it is very difficult to explain why this method, this protein prediction is very high, why this accuracy is low, and so on. Summarizing, we discussed on various methods, right. What are the various methods we discussed? Right. First one is the propensity, right. So, or we can say Chowan-Passman, and we discussed about information theory, GAR here, right, GOR. Then we put the hydrophobicity profiles, here we can predict with the single values as well as with the average windows, right. And then we discussed about the multiple sequence alignment, right, MSA. Here also you have two ways you can assess the multiple sequence alignment, either take the parameters from the GOR, or you can get the information from the experimental data. So, you can predict the secondary structures, right. So, also they found that if the alignment grows highly conserved, then the prediction performance also increases. So, JPROD is the one, or ZPROD, they first used multiple sequence alignment for predicting the secondary structures, and then we discussed about the neural networks, right, or you can see the machine learning. Right, here they take the input information and they try to predict the output, right, that is a secondary structure. Then finally, we discussed about the consensus. Okay, two types of consensus methods. One is based on ensembles. The ensemble based method, how it works?
Majority voting.
Majority of voting. They take the several methods, right, and get the voting, which one has the highest one. So that is a secondary structure, helix or strand or coil, right. In the case of the meta predictions, what they do? They train the output of each single method and finally use the information for predicting the desired output, right. This is how they do. So now at the present scenario, right, you can see the variations of the performance, and we could see about 75 to 80% accuracy, right. Now we can predict the secondary structures. So currently, okay, due to the advancements in the computing, right, say the big data with large amount of data and deep learning, they are trying to use deep learning in almost all prediction purposes, including protein secondary structure prediction, right. They are trying to map this information, right, and then try to improve the prediction performance. So we discussed about the secondary structures. In the next classes, we will go through the tertiary structures, right. So what are the major information we obtain from protein 3D structures, right, what are the various databases and how can we use the information available in protein 3D structures, right, to derive any properties, and how can we use these properties with the different applications for the predictions or how to understand the folding and the binding and so on. Okay, thanks for your kind attention.